Welcome to this tutorial video. So in today's session, we are going to continue looking at viruses. So make sure that before you watch this video, you watch the part 1 video that I had uploaded on viruses. So in the first video, I'll just brief you on what we talked about. So in the first video of part 1, I introduced the topic on viruses, where we described the virus, we defined what a virus is. Then we also went further to look at the terms that we use in the description of a virus. Okay, we did that in the part 1 video. Apart from that, we also went further to look at the features, or rather the properties that distinguish a virus from other living organisms. Remember, we said a virus is not a living organism. It's basically a protein. It's basically a set of DNA that invades different body cells and then causes a disease or an infection. Now, in today's session, I want to talk about very important things, and one of them is the classifications of viruses. So as a student that is in the field of medicine or medical science, then you've got these microbiology course, and then you're looking on viruses. You have to understand how these different viruses are classified. Remember, these classifications, they help you in being able to find the specific or best approach in how to treat a specific disease that is caused by a virus. Apart from that, I also try by all means to reach as far as talking about the replication cycle of a virus, because a virus is not a living thing. Then you must understand to say, how does it divide? How does it increase in number? How does it multiply? So that's what we're going to look at. So before we continue, remember to subscribe to this YouTube channel. As you know, it's been providing you with the content that you need, especially if you're doing programs such as Bachelor of Science in Nursing, Bachelor of Science in Midwifery, registered nursing, registered midwifery, and the likes. So let's continue. Now, it's very important that even before we proceed, we get to look at what we refer to as the anatomy or structure of a virus. Now, it's very important for you to know that viruses can either be enveloped with a lipid membrane around the cuspid. What I mean is that these viruses that we're talking about, as we talk about the structure of the anatomy, you hear something like a virus is enveloped, meaning it's closed into something. So when we say a virus is enveloped, it simply means that around the cuspid, I'm going to show you very soon, there is a lipid membrane, the same lipid barrier that you know. Okay, there is a lipid membrane that is surrounding that that particular virus, or surrounding what we call a cuspid. So we're going to call it an enveloped virus. Now, there is also what we refer to as a naked virus. So a naked virus is that which is not enveloped. It simply means that it doesn't have the outer membrane. So we refer to it as what? As a naked virus. It simply means that around that cuspid, there is no that lipid membrane. So it's going to be referred to as a naked virus. Apart from that, knowing the structure of a virus, there is also what we refer to as, we call them spikes. So we're saying that viruses often have surface like protrusions. Okay, so they have got what we call spikes. I'm going to show you very soon. These spikes are used for attachment to the host cell. Remember, a virus has to attach to the host cell for it to be able to cause an infection. So how does it attach to the host? It uses what we refer to as the spike. So we're saying that the spikes are used for attachments to what? To the host. Apart from that, looking at the structure or the anatomy of a virus, you have to know that the basic shapes, so these viruses that exist, the COVID-19, the HIV, the influenza virus, and the likes, what you must know is that they are in different shapes, okay? They possess different shapes, or they assume different shapes. One of the shapes is what referred to as spherical. I'm going to show you very soon, okay? There's what referred to as spherical or icosahedral shape. Apart from that, there's also what we call as cylindrical, meaning it's a rod shaped like. I'm going to show you these structures very soon. As a student that is, studying microbiology, you must know that a virus, no matter the type of virus that you are talking about, a virus can only be seen using what we refer to as an electronic microscope. As you are aware that there are different types of microscope that exist. There's what we refer to as a light microscope, compound microscope, pocket microscope, different types of microscopes. But if you're talking about a virus, if you want to visualize it under a microscope, you can only use what we refer to as an electronic microscope. And I must remind you that this question, they like it in an exam. They'll ask you to say what type of microscope can be used to visualize a virus. You tell them to say it can only be an electronic microscope. So what have we talked about on this particular slide? We introduced the anatomy of a virus. How does a virus look? What is its structure? We've said that a virus can either be enveloped, it simply means that it is enclosed or it's protected into a lipid membrane protecting the capsid. And then we've also said that this virus can also be what? Naked. It simply means that that lipid membrane is not there. Then we've said that a virus also uses what we call spikes to be able to attach itself to the host and cause an infection. Then we've talked about the basic shapes of a virus, which can be spherical, cylindrical, or rod-shaped. And then we've said that indeed a virus can only be seen by using an electronic microscope. So this is what I was talking about. So you can see that when we say that a virus is enveloped, it simply means that it has got this lipid membrane around it that is surrounding the capsid, because this is the capsid that you're seeing inside there. Then it's protected by this lipid barrier membrane that is outside here, okay, in blue light. So it's protected. So I'm going to say it's enveloped because it's being protected. Remember, when you put a letter in an envelope, you are protecting the letter inside, right? So we're going to say this type of a virus is what is enveloped. Okay. Then I also talked about the spikes. So these are what referred to as the spikes. These protrusions that you are seeing here, these we call them spikes. So these spikes, like I've mentioned, they are used in helping a virus to be able to attach to a host. So how does a virus attach to your blood vessel, or to your blood, or to your skin, or to your body tissues? It uses what referred to as the spikes. They are the ones that are going to stick to your different body tissues, and then they're going to cause an infection. Then I've also said that a virus can also be what? Naked. If it's naked, you can see that this outer membrane bilayer is not there. This envelope is not there. It's naked. So it's directly linked to the spikes. You've got the capsid here that is directly linked to the what? To the spikes. Of course, you can see that inside this virus there is what? Nucleic acid. Remember, I've always emphasized that a virus is simply a set of genetic material. It's basically a set of proteins. It's not a living organism. So it simply means that inside this, there should be what referred to as nucleic acid, or its DNA, which can be either RNA or DNA. So that is what I wanted you to know about the anatomy of the virus. So talking about the specific things which I've already talked about. So we're just explaining now these different entities that you are seeing on this shape. So for example, we're saying that nucleic acid core, what do you find inside there? We're saying that in the nucleic acid core, they can either be DNA, deoxyribonucleic acid. So inside this capsid, you can find this virus can contain DNA, others they contain what you call RNA. So a virus is made in such a way that other different types of viruses can have RNA inside the genetic material or genome. Others are going to have DNA. So they are in different forms. So we're saying that inside this genome, they can either be what? DNA or RNA. They can either be single or double-stranded, which carries the viral genetic information. I'm going to talk about single and double-stranded very soon. For now, I just want you to know that inside the nucleic acid core, you can find DNA or RNA in this particular virus. Then a capsid, we're saying that a capsid is basically a protein shell that encases the nucleic acid made up of subunits called the caspomeres. Okay, so what we're saying is that this particular capsid is just enclosing this particular, you can see, this capsid here is basically a protein that is enclosing the nucleic acid core. Okay, you can find that inside this, that's why now you find it can either be DNA or RNA, and this capsid is the one that is enclosing this particular nucleic acid core. That's what we are saying there. Okay, so I think that the other part that we talked about on the anatomy, we said the virus can have an envelope. It's optional. It's either this virus can have an envelope, which we say an enveloped virus, or it can be naked, right? So we're saying that some viruses possess a lipid envelope derived from the host membrane. Imagine it's getting this particular bilayer from you as a host. If it infects you, it will make sure that it wears that membrane so that it's well protected. So we're saying that some viruses possess a lipid envelope derived from the host membrane embedded with what? Viral proteins. A virus is basically a set of proteins, like I've always said. Okay? Then apart from that, we talked about the surface proteins, or what we refer to as spikes. I said they facilitate attachment and entry into the host cell. How does it get to enter your cells? How does it get to cause a disease? It's going to use these different spikes that I've talked about to be able to invade your cells, and then it will be able to cause what? An infection. And like I said, on the basic shapes of the virus, I said they can either be what? They can either be helical. So helical, you can see that these are basically cylindrical. This is more like a cylinder, right? So they can either be of this shape. Okay? They can be of this shape. Then apart from that, they can also be what? Spherical or icosahedral shape. I talked about that. Then they can also be what? Complex. If you went further in microbiology, there's what we call bacterial genetics, where there's a component which we refer to as a bacteriophage. A bacteriophage is basically a virus that is used to transmit some set of DNA from one bacteria to another. It's usually it usually possesses what we call a complex shape. It looks like this. Okay, so what I just want my students or the people that are watching this video to know is that a virus can have different shapes. It can have a helical shape like a cylinder, as you can see. It can have a spherical shape, round like, or it can be complex, as you can see. It's looking like a machine. Different kind of parts or elements on this particular shape. So those are the shapes of a virus. Now, moving and talking about classifications of a virus. Now, you've always known that in medical science or in medical field, whenever we get to classify microorganisms, or whenever we get to classify these different bacteria or viruses, that particular classification is very important. Why? Because it's the one that guides us on the treatment. So if we classify this virus, we'll be able to identify to say if this virus is like this, then how do we go about treating it? Is it spherical? Is it a DNA virus? Is it a double-stranded virus? Is it a single-stranded virus? So these classifications are not for decorations. They are not for entertainment at all. In medical science, we use them to find a better approach to help a patient that has been infected by that particular virus. So we're saying that viruses are classified based on several fundamental characteristics which include, so we're saying that these viruses, we classify them based on different aspects. Number one, we can classify a virus depending on the type of nucleic acid that it contains. So it can either contain DNA or RNA. Okay? So it simply means that there will be what referred to as DNA viruses and what referred to as RNA viruses. For example, hepatitis B is a DNA virus. Okay? If you're talking about HIV and AIDS, that is an RNA virus. So we can classify them depending on the type of nucleic acid that they contain. Number two, we can classify them depending on the strands. So the strands, we're talking about the genetic material that they have. This DNA or this RNA, is it a single strand? Is it a single rope of genetic information? So we're saying that these viruses can either be single-stranded. You will see this, even in an exam, they're going to use this abbreviation as SS. It simply means this is a single-stranded virus. Or it can be double-stranded, abbreviated as what? DS. So we're also saying that we can classify viruses depending on the strands. They can either be single-stranded or double-stranded viruses. Apart from that, there is what we refer to as RNA sense. This is very important for a medical student. Either nursing, midwifery, or whatever program that you are doing, as long as you've got this course of microbiology. When we say sense, so you hear things like this particular virus has got an RNA sense, or a positive RNA sense or a negative RNA sense. It can either be positive sense or negative sense. Now, I want to explain something here. When we say this virus has got a positive sense, what it means is that, remember I said a virus is simply a protein. It's simply a collection of genetic material that is full of proteins. So it simply means that if a virus is positive sense, what it means is that its RNA, its RNA material can be translated directly to proteins. Remember when I introduced this topic, I talked about the central dogma. That is the movement from DNA to RNA and from RNA to what? To proteins. From DNA to RNA, that is transcription, right? Then from RNA to translation, okay, or to proteins, that is what we call as it translation. So when we say a virus is positive sense, it simply means that we can directly translate it from RNA directly into what? Into proteins without it having to be what? To be transcribed. Then when you say if it's negative sense, it simply means that it can't go to form proteins directly, meaning you must reread again. It must be transcribed again. That's what we mean. So these viruses can either be positive sense. Whenever you see that a virus is positive sense, it's very dangerous. Why? Because it can easily form proteins, and proteins are the actual viruses that viruses exist in. They exist as proteins. A negative sense is a bit, not severe, but positive sense viruses are very, very bad because they can easily be transcribed into proteins. Then apart from that, they can also be classified based on the shape or the symmetry. How do they look like? Like I mentioned, they can either be cubic, they can be helical, or they can be complex. You can see there are different forms, different ways in which we can classify these viruses that we're talking about. Apart from that, they can also be classified based on the replication strategy. How do they replicate? How do they double, in short? How do they multiply? So we are saying that replication strategy, we are saying that a virus can either be what we refer to as, there's what we call including reverse transcription in retroviruses. So most of the times you've heard people talking about HIV, talking about the drugs that are used in treatment of HIV, ARVs, right? Antiretroviruses, that's the name, right? So I'll explain what it means when we say a virus is literal. So it's a very, very important concept because you are aware that even in the very day that you are living in, we've not been able to find the cure to be able to fight that virus that causes what? HIV. People are just given drugs that are used to manage it because it's a literal virus. Literal viruses are very difficult to deal with. So that is what I wanted you to understand on the fundamental classifications of virus. So when you hear something like single-stranded, positive sense, negative sense, helical, you are familiar with those terms. They are just used to classify because these viruses, they exist in different forms. Recently, you heard of COVID-19, Ebola virus. They are different form of viruses. Okay, so let's proceed and see what we are supposed to look at next. Okay, now this is what I was talking about here. Okay, I think I've already mentioned on what, when we say single-stranded or double-stranded or positive sense or negative sense, what they mean. To give you a picture or a view, let's take for example, you can see that on picture number 1, we've got what we call DS. I said DS means what? Double-stranded. So if it's DS, if it's double-stranded, it's DNA. DNA is the only kind of genetic material, but it's double-stranded. RNA is always single-stranded. So double-stranded, it's double-stranded, it's DNA, meaning this one, it's supposed to be transcribed because you can't form proteins when it's in form of DNA. You must transcribe DNA into RNA. From RNA, that's when you move to be able to form what? Proteins using translation. So this one can move from DNA to mRNA. Then apart from that, you can see on this particular end, we've got what we call single-stranded, okay? Single-stranded positive sense. It's single-stranded positive sense. So what it means that if it's a positive sense virus, okay, it simply means that it can be transcribed directly into proteins because it's already an RNA. So this one can go to form a virus, an actual virus directly, because when you're forming proteins, if you're talking about viruses, you are actually forming the actual virus. That's how it works. Okay? Then just to talk about the most important thing, let me focus on this part, number 6. So number 6 is talking about literal viruses. And you are well aware that HIV viruses are literal viruses. In case you don't know, the most important thing why you learn about viruses in microbiology is that they want you to understand how the HIV virus works because it's the most difficult one to deal with. So an HIV virus is a single-stranded RNA virus. Now, what you must know is that it's a literal virus. What does it mean? The way the HIV virus works is like this. Instead of moving from an RNA, because it's already RNA, right? So instead of moving from RNA, going to form the proteins directly, when I'm saying it's forming proteins, it simply means it's forming the actual virus, because the virus is a protein. So what the HIV virus does is this. It knows that it wants to be able to cause very severe harm to you. So what it's going to do is this. Instead of moving direct from the RNA to form the actual protein or the actual virus, it's like, okay, I'm going to go back to DNA. So it undergoes what we refer to as the reverse transcription. No wonder we call it retrovirus. So it's going to move from RNA going back to DNA. So that is what we refer to as what? Reverse transcription. If you ask yourself as a medical student, why is it going back to DNA? It simply means that from DNA can what? DNA can double. It can replicate. So this virus, if it goes from RNA, it goes back to DNA, it's going to multiply. It's going to duplicate. The DNA replication, in short. So it's going to increase in what? In number. Then from that now, that's when it's going to go at it's going to go to what? To mRNA now, and then form proteins. In short, the overall goal why it's a retrovirus or why it's performing reverse transcription is that it wants to increase in number. If it was just one here, if it goes back to DNA, there'll be 10 of them, there'll be 15 of them, or there'll be even 20 or 100, because it has gone back to DNA. Once they do that, then they now go back to the actual mRNA. And you are well aware that this mRNA now can be used to form the proteins, which are the actual viruses in this particular context. So that's what I wanted to explain on this particular slide. Okay. Now, this table that I'm showing you, it's not very it's not a must that you must master everything, but I'm just showing you that in case you come across this set of classifications, you can see that we are classifying viruses based on the family, the actual virus, the disease that they cause, the nucleic acid, the shape, the RNA sense, and the strand. For example, if we're talking about retroviruses, I'll just talk about retroviruses, for example. So retroviruses, they fall under, that's the family, right? Retroviruses, that's the actual family. Remember, if we're talking about the taxonomy of microorganisms, we move from those different domains, from family, species, the genus, and everything. Okay, so the family, for example, we're talking about retroviruses, right? The actual name would be the HIV virus. So the HIV virus is falling under the family of retroviruses. What disease does it cause? It causes disease such as AIDS, okay, T-sickle cell, T-cell leukemia. I don't know if you've heard of it. Okay, it's basically the cancer of the blood, right? It's induced by viruses, okay? So it's basically some autoimmune disorders like and the likes. So these viruses can cause AIDS, or they can cause what we call leukemia. What type of nucleic acid do they contain? HIV viruses are always RNA viruses. Okay? What kind of shape do they have? They've got a helical shape, a cylindrical shape. I've shown you. You've seen, right? That's why I was showing those different shapes. Okay? What type of RNA do they have? Is it a positive sense or a negative sense? Okay? Positive sense with the reverse transcription. So they are positive sense, meaning they can move directly from RNA to form proteins. And I said they're very dangerous because a positive sense virus is very dangerous. Why? Its its speed of multiplying or increasing number is very high because it doesn't have to go back to RNA. It moves directly from its RNA to form proteins. To make it worse, an HIV virus is a retrovirus, meaning it can go back to DNA using what we call as reverse transcription. When it goes back to DNA, you know what DNA can do? It can increase in number, it can double, it can replicate. Then they are going to increase in your body. Then to be able to, decrease what we call the CD4 cells, which we're going to talk about very soon. Apart from that, you can see that it's single-stranded. It's automatic. You don't have to stress as a student. If a virus is an RNA virus, why must you stress if it's double-stranded? Of course, you already know that it's single-stranded. So these are the ways in which we classify viruses based on the family, the actual virus, the disease that they cause, the nucleic acid, the shape, the RNA sense. Is it positive sense or negative sense? The strand, okay? Remember I said, if it's positive sense, it can form proteins directly. Direct. If it's negative sense, it can't form proteins directly. It has to go back to RNA. Okay? That's what it means. So this is the table that you can use just for references. Now, importantly, we have to talk about the virus replication cycle or the reproduction mechanism. So if you ask yourself, I say that a virus is not an actual living organism. It doesn't have the organelles. It has no mitochondria, it has no Golgi apparatus, it has no ribosomes. If you ask yourself as a medical student to say, but how is it that these viruses they cause harm to individuals, to the general public? Is that these viruses they use the host machinery. What I mean is that a virus cannot survive on its own. It always needs to be in somebody's cells. It can't survive on its own. So I'll just introduce this particular concept of the virus replication mechanism. Then I'm going to make an actual video, a single video that is talking about the actual replication cycle of the virus. I'll just introduce it right now. I'll just introduce the concept right now. So I'm saying that this particular virus, okay, it uses the host cells. It simply means that on its own it can't survive. This is why it's very difficult for a virus to survive outside a living host, because it's not a living organism. It's not like a bacteria. On its own, it cannot survive. So the picture that you are seeing here is a brief summary of the virus replication cycle or the reproduction mechanism. Okay, there's what we call attachment, endocytosis, integration, all those different concepts and budding and the stuff. I'm going to talk about them. I know you're waiting for me to talk about entry, fusion, and those kind of stuffs. We're going to talk about that. Okay, and then I think I've already talked about the viral replication mechanism. I was saying that the viral replication mechanism, this refers to the process by which viruses reproduce new virions. When we say virion, a virion is an actual intervirus particle that can cause a disease. I defined this in my first video. On that said, if you didn't watch the first video, make sure you go back and watch the first video, then come and watch this one. So viral replication, this refers to the process by which viruses produce new virions, or they produce other viruses within a host cell. Okay, so viruses are entirely dependent on the host cellular machinery for what? For replication. What it simply means is this: a virus, for it to multiply, for it to double in number, for it to increase, it can only do that if it's inside you as a human being, okay, or if it's inside an animal. So on its own, it can't multiply. On its own, it can't, increase in number. So it must first enter you, whether it's your blood, okay, whether it's your tissues and the likes. Now, it's very, very important for you to know what we refer to as the CD4 cells. I'm introducing these concepts because in my next video, I'll just go directly into explaining the actual virus replication mechanism. So they are what we call as CD4 cells. Now, on my videos on immunity, I think I did explain what CD4 cells are, but I'm going to repeat. CD4 cells simply means cluster differentiation type 4 cells. Now, these cells are type of white blood cells. They are an element of the immune system. Okay, remember, we're talking about the immune system. This is a system that helps you and I to be able to fight, diseases or infections, right? Now, what actually fights at molecular level are the cells. So, we know that white blood cells are the ones that are used in fighting microorganisms. So, these CD4 cells, there are specific types of white blood cells. I'll not go into details. There are specific types of white blood cells. So, the CD4 cells, what do? They are sometimes known as T helper cells. Maybe you've heard of them. There's what we call T helper cells type 1, type 2. But don't have to worry about that. All you have to know is that the CD4 cells, they are the ones that help you to fight different infections. If they ask you, how do they help you? They activate what we call antibodies. Antibodies are the actual soldiers of the body that go to fight those microorganisms that infect you. It can be bacteria, it can be virus, a protozoa, or a fungi. So what these viruses do is that they understand to say, if I infect this human being, if I infect them, the CD4 cells are going to fight me, because once they notice that I'm inside the body, they're going to send the what? They're going to send the antibodies and they're going to kill me. So what do the virus do? They are going to target the actual CD4 cells such that when they infect the CD4 cells, they are going to kill them. So there will be nothing that they're going to send to be able to fight them. So that's how it works. So the virus targets what we call the CD4 cells, cluster differentiation cells type 4. Some of you've heard the things such as, the CD4 count cell is low, especially in people who have got HIV and AIDS. It simply means that the viral load has increased. The more the viral load has increased, the less number of the CD4 cells. And the less the number of CD4 cells, the more somebody's immune system is going to be compromised, because they're going to be weak. the CD4 cells are the ones that help them to activate antibodies. Antibodies, they go to fight those different infections. But if these CD4 cells have been reduced in number, then you can't be helped. So the virus makes sure that it infects these CD4 cells, cluster differentiation cell type 4. So I'm going to talk about, if I start talking about the actual viral replication mechanism, I'll be talking about the CD4 cells, oftenly. Okay? So this is the viral replication mechanism or reproduction mechanism. But I'm going to talk about it in the next video in details, where, as you can see, I'll start talking about the actual replication cycle. Now, in this particular context, I'm going to use the HIV virus as for example, because it's the one that we have been combating for many, many years now. So instead of talking about any other type of virus, I'm going to use the HIV virus to make you understand how it multiplies. How does the HIV virus increase inside the host cell? How does it increase in number? How does it get to reduce the CD4 cells? How does it get to increase the viral load? I'm going to explain in the next video, where I'll talk about the binding or attachment phase, the fusion, reverse transcription, integration, replication, assembly, and the budding off or maturation. I'll remind you, ensure that you subscribe to this YouTube channel so that you don't miss out on that particular concept. For now, we're going to end on this particular part.